This particular grant is focused on the salt springs of Manitoba, and it's a really special environment where we've got these clearly separated springs right here in our backyard. Many Manitobans don't even know that we have these salt flats and salt springs in Manitoba, and what makes them so unique is that they're this far separated geographically from anything that is similar in terms of their chemistry globally. It means that the biodiversity that inhabit these springs have basically evolved completely separately from other populations globally. And it means that there's a very high level of novelty there. So there's a bunch of new microbes that we've never even discovered before, and this is the first time that we're actually able to encounter them, and they're completely new to science. So this obviously means that there are very serious considerations to take into account when we're thinking about preserving that biodiversity. Currently, my lab is focused on three main topics. I would say the first is associated with discovering this new microbial biodiversity that we haven't been able to study before. And the second theme would then be associated with providing the language necessary to communicate effectively about these organisms. So names that mean something and that imply something about the evolutionary history of the organisms themselves. And then the third main theme is associated with leveraging some of those microbial physiologies to solve societal problems. So my Research Manitoba grant is actually allowing us to better understand this process of speciation. And it's actually the mechanism that drives the generation of this biodiversity in itself. And if we're thinking about what those microbes evolve towards, how they look different in these different environments. So obviously, geographically, they're completely separated from other springs, but it also means that they start evolving on their own diverging trajectories in the different springs. And as part of that exploration process in my lab, we're trying to understand the mechanisms that are providing this action. So at the same time, if we can actually understand the breadth of microbial biodiversity, it can give us an indication of how many of those microbes could be harmful to humans or might be beneficial in being able to perform certain societal functions for us. So we're typically operating on the assumption that all microbes would survive, or if they don't survive, maybe they don't need to survive. But this might be a flawed assumption, and we need to understand this diversity in order to preserve it better. So what gives us a better indication of how they evolve would ultimately drive our biodiversity conservation strategies and policies and have an influence in terms of how we're looking at the biodiversity in Manitoba itself. And if we get to a point that we can leverage these organisms for solving societal problems, it can actually bring real life contributions to Manitobans that we see. And this could even be in the context of agriculture, where we can understand the different crops better, get a better idea of what the natural biodiversity for crops need to look like, and how human influence is actually altering that biodiversity. And even when we're thinking about things like antimicrobial resistance mechanisms, those are naturally present in the environment, but if we don't understand the breadth at which that is present in the environment, we don't necessarily understand how we're going towards monitoring it. So even though this grant is focused on like a very fundamental research, foundational research principle, we need to understand these principles in order to get to a point of understanding the real-life application-based implications for this.